This is the second of two videos about phototransduction, the first step in vision. In part 1, we looked at the process of phototransduction as it occurs in rod cells in the retina. We got as far as photon arrival and activation of the nerve signal. The doorbell had been rung. In part 2, we will go on to talk about how the signal is turned off. You want the doorbell to ring once, not keep ringing. We will see how the retinal is regenerated, some technical details, and some differences between rods and cones. Let us start with a brief recap of what happened in part 1. We discussed that phototransduction occurs in the outer segment of the rod cell. In the dark, the low level of guanylate cyclase activity results in enough cyclic GMP available to keep the CNG channel open, and that sets the resting charge of the photoreceptor membrane. When light arrives, rhodopsin is the receptor molecule. Incoming light is absorbed by the embedded retinal, which causes it to change shape, which in turn affects the shape of the opsin molecule, causing it to take its active form, meta-rhodopsin 2. The active rhodopsin then activates intermediate transducin, which activates phosphodiesterase, which then breaks down cyclic GMP molecules. As the amount of cyclic GMP is reduced, the CNG channel closes and the internal cell charge becomes more negative. That finally results in a change in glutamate secretion at the synapse with the bipolar cell. The switch is turned on, signaling that light has arrived. The doorbell has been rung. Now, once the system has been turned on, it is important that it be turned off just as rapidly. To use our previous analogy, you don't want the doorbell to continue to ring once it's been pushed. The principal parts of deactivation occur in two places. First, to deactivate the rhodopsin. The first thing that happens is G-rhodopsin kinase attaches to the rhodopsin and begins to add phosphate groups, up to 3. With each phosphorylation, rhodopsin activity decreases. Second is attachment of the protein arrestin. This stops all rhodopsin activity. It is no longer able to act on the transducin. Soon after, the spent retinal in the all-trans form is dropped away from the opsin molecule. We will follow retinal regeneration later on. For now, we're just turning the system off. The second deactivation step involves transducin. The transducin was initially activated by changing GDP to GTP in the alpha subunit. Beta and gamma subunits separated, leaving active transducin alpha. To deactivate the transducin requires changing the GTP back to GDP. This would occur very slowly if the transducin were left alone, but it has help. There is an enzyme, regulator of G protein signaling, RGS for short, that assists the rapid hydrolysis of GTP back to GDP, thus inactivating the alpha subunit. The alpha subunit then rejoins the beta and gamma subunits, again forming the inactive transducin complex. The RGS complex has 3 parts. First, RGS9-1, that is the 9th member of a family of GTPase accelerating proteins. Consistently partnered with this is G-beta 5, and R9 anchoring protein anchors the complex to the cell membrane. Altogether, this is known as a gap complex, and its action may be the rate-limiting step in controlling rod recovery speed. If any one of the parts of this complex is missing, rod recovery time is 10 times slower. The activated transducin was responsible for restraining the gamma controllers of phosphodiesterase. As the transducin becomes inactive, the gamma controller subunits of PDE again take effect, and the PDE returns to its minimally active resting state. With the PDE less active, cyclic GMP begins to reaccumulate. The CNG channel reopens, sodium and calcium ions begin crossing into the cell, and the electrical charge returns to the resting level. The doorbell has been turned off at 1 ring. While light is the trigger for phototransduction, calcium is a key ion in controlling recovery events, so let's specifically review its role. Phototransduction and membrane charge are based on a balance between guanylate cyclase activity making cyclic GMP and phosphodiesterase digesting cyclic GMP. The presence of cyclic GMP keeps the CNG channel open for ions to enter the cell, helping regulate its charge. That is one big role for calcium ions, but there are more. Recall guanylate cyclase activating protein, GCAP, which regulates guanylate cyclase activity. In the dark, calcium level is higher, which tells the GCAP to inhibit guanylate cyclase activity, keeping it low, but not zero. Some cyclic GMP is constantly being produced. In the light, calcium level drops, and this tells the GCAP to activate guanylate cyclase, producing more cyclic GMP, causing the CNG channels to open. Calmodulin, abbreviated CAM, is another regulating protein. It is attached to the CNG channel. When calcium level is high, it reduces the sensitivity of the CNG channel to cyclic GMP, leaving the channel less open than it would otherwise be. When calcium is low, it allows for increased sensitivity of the CNG channel to cyclic GMP, allowing more ions to pass. The third calcium-controlled regulating protein is recoverin. It controls the lifetime of activated rhodopsin by its action on G-rhodopsin kinase. In the dark, when calcium is high, it inhibits G-rhodopsin kinase. In the light, when calcium decreases, it releases GRK inhibition, which allows for phosphorylation of the rhodopsin, helping to turn it off. Low rhodopsin means less PDE, more cyclic GMP, and a rise in calcium level. There is more to the recoverin story, but we will not be following that at the moment. You see the overall strategy here. When light arrives, the process of phototransduction causes the CNG channel to close. The calcium level would stay the same, but the NCKX exchanger continues to pump calcium out, so the concentration drops. When the calcium level drops, it is the aim of the calcium-based regulators, GCAP, calmodulin, and recoverin, to help restore the calcium level, and to do so rapidly to be ready for the next photon. So far, the system has set its resting potential in the dark, been activated by the arrival of light, and then rapidly deactivated. Here the transducin and phosphodiesterase are back in their resting states. But the retinal has been used, that is, changed to the all-trans form, and needs to be changed back to the cis form in order to be ready for more photon arrivals. This is how the cis retinal is regenerated in rods. It is called the visual cycle, and this will be a fair journey on its own. Phototransduction occurs in the photoreceptor outer segment. Transmission of electrical impulse occurs through the synapse. The visual cycle, regeneration of retinal, occurs in a separate cell, the RPE cell. Here is our 11-cis retinal, free, unbonded, in its familiar shape, bent at the 11-12 double bond. Retinal because it is an aldehyde. Note the double-bonded oxygen. The retinal binds to the opsin protein via this nitrogen in a lysine residue at position 296. When light is absorbed by the 11-12 double bond, it allows the carbon chain to straighten out into the trans form. This quickly leads to the active form of rhodopsin we talked about before. Soon the retinal is disconnected from the opsin, briefly existing as free-floating all-trans retinal, which has significant toxicity to the cell. It is quickly turned into all-trans retinol, the OH group making it an alcohol. The all-trans retinol has gone as far as it can within the photoreceptor cell, For the next steps, the retinal must cross the space between the photoreceptor and the RPE cell like crossing a moat. There is no drawbridge, but there is a binding protein to help it across. Once on the RPE side, a side chain is attached. This is a retinal ester, which then becomes part of a storage pool within the RPE cell. At some point, the retinal ester is grabbed from the storage pool and changed into 11-cis retinol. And one more simple step and it is back to 11-cis retinal and ready for action, except that it is still on the RPE side of the moat. Again, it gets its binding protein to help it back to the photoreceptor cell. When needed, it binds to the opsin protein and is again set and ready to receive light. Here is an interesting point. In invertebrate photoreceptors, the all-trans retinal is reset to the 11-cis form when another photon arrives. It does not have to travel through the whole process we just described. You have now seen the whole process of phototransduction and the visual cycle. Was it not amazing? Decades of time and effort were required to map out all of those steps. Now that we have covered the individual steps, we look at how they fit into a broader picture. The energy of a single photon is tiny. How can it be amplified enough to reach a threshold that can register on our biologic molecular size sensors? There is also the important subject of light adaptation, but that is beyond the scope of this presentation. Maybe another day. An important concept to understand is how the signal is amplified. That is the idea of being able to detect and signal the arrival of a single photon. Here's how that works. First, one photon activates a single molecule of rhodopsin. Second, a single activated rhodopsin then activates about 400 G proteins per second. Third, the G proteins then activate phosphodiesterases, 1 alpha subunit per 1 gamma subunit. PDE is a very efficient enzyme, hydrolyzing cyclic GMP at a very rapid rate, 2,000 or more per second. Fourth, the CNG channels close. The sensitivity of the CNG channel is such that when cyclic GMP concentration has dropped to half, the channels remaining open drop to about 1/8. Overall, the capture of a single photon causes a reduction of 2 to 5% in the CNG current. That causes a change in membrane charge of about 1 millivolt. As a practical matter, we can ask, how fast do we recover vision when going from bright outdoors to darkness indoors? That can be formally tested by timing how fast the retina can recover light sensitivity after bleaching by a bright light. This graph from the 1975 paper by Pugh shows how vision recovers after light exposure. After a bright exposure, the initial recovery involves the cones, which recover relatively rapidly, in minutes, but to a limited sensitivity, whereas the rods recover more slowly, tens of minutes, but eventually reach a more sensitive threshold. Note the left scale is in log units, which nicely leads to our next subject. So far, we have been discussing exclusively how rods work. Before we finish, let us at least visit the world of the less numerous but more important cone cells. Rods outnumber cones about 20 to 1. The intention of this section is not to review cone function and color vision in detail, it is just to highlight the differences. Color vision is covered in a separate set of videos. Here is a question for you. In the evolution of the eye, which came first, rods or cones? The answer is cones first, rods came later. It makes sense that organisms learn to function in light first, though it can be proved in other ways. The first visual pigment was sensitive to long wavelengths, casually called a red cone. The second pigment was sensitive to short wavelengths in the UV violet range, called a blue cone. Interestingly, after some additions and subtractions over time, this is what most mammals have ended up with, red and blue cones, in addition to rods. More recently, duplication and mutation of the long wavelength cone ended up creating the middle wavelength or green cone. 3 color cones is the color complement we humans and some primates have now. Rods sprang from the short wavelength cone pigment. This is covered in more detail in the videos on color vision. Here are the peak sensitivities for each of the cone types, and here is where rods fit in. So cones give us color vision and work in bright light. Rods, on the other hand, work in dim light. They are so sensitive they can count single photons, which is amazing if you think about it. They do not in general interact with the color system. They give us a grayscale vision. To give you an example, a single photon response in rods is about 1 millivolt, a few percent of the maximum rod response, with a time to peak response of 200 milliseconds. Cone response is faster, peaking at 30 milliseconds, but their response to a single photon is smaller, too small to separate from background noise. Structurally, the rod outer segment has the disks we talked about before that are contained entirely inside the cell. This schematic drawing shows that cones are surprisingly different. They have indentations or folds in the cell membrane, not the internal disks. The retinal, which detects the light, is the same molecule in rods and cones. It is the opsin molecule that differs. Difference of a few amino acids results in a slight difference in how the opsin molecule holds the retinal, which is what determines the wavelength of light it is most sensitive to. The next item is also somewhat of a surprise. The visual cycle used by the rods is shared by the cones. Okay. However, cones have an additional visual cycle that runs through the Mueller cells in the retina. We did not discuss Mueller cells further here, except to say that they are glial cells important to several retinal functions. In addition to the different location, there are also different enzymes involved in the cone-dedicated visual cycle. Proposed enzymes are listed in parentheses. To recap rods versus cones, rod attributes are on the top, cone below. We covered significant differences in structure and sensitivity to light and color. Differences within the G protein cascade are listed here. The opsin difference means the GP coupled receptor differs between each of the 4 cell types. Likewise, the G protein subunits are closely related, but are not exactly the same. The effector molecule, PDE, in cones exists only as an alpha subunit. The CNG channel has different alpha and beta subunits and combinations. The exchanger is different. And, as we mentioned a minute ago, there is one visual cycle that are shared by rods and cones, but there is a second one that is used exclusively by cones, which may help in their ability to recycle faster. With that summary of rods versus cones, we come to the end of our exploration of phototransduction. Now, let us conclude by taking a direct run through the whole process. The phototransduction cascade is a G-protein coupled receptor system. It shares the same process that works in many kinds of cells, for example, cells that respond to hormones and cells that respond to taste and smell. The receptor is rhodopsin, the G protein is transducin, and the effector protein is phosphodiesterase. Resting potential is set in the dark by a low level of guanylate cyclase activity, producing cyclic GMP, which keeps the CNG channel open. That allows sodium and calcium to enter the cell and keep the membrane charge at a resting potential that is below its full potential. When light arrives, it activates the G protein cascade, starting with rhodopsin, which acts on the transducin and phosphodiesterase. That reduces the level of sodium and calcium coming into the cell, which makes the cell more negative. In the dark, the resting potential is relatively lower, and the cell is producing more glutamate at its synapse. When light arrives, the resting potential becomes more negative, and the cell produces less glutamate at its synapse. When enough light arrives and all the CNG channels close, the cell stops producing glutamate. That is how the cell signals dark versus light. Just as we needed the system to turn on rapidly, it also has to turn off rapidly. Deactivation of rhodopsin requires several steps of phosphorylation, followed by binding of an arrestin protein to fully stop its activity. The activated transducin was deactivated with the help of the gap complex. With transducin inactive, that again slowed down phosphodiesterase activity. With PDE activity slowed, then cyclic GMP could accumulate, and the CNG channel again opens up. The cell membrane charge then drops back to its resting potential, ready for another photon. And that is the cascade of phototransduction. The level of calcium within the cell plays a significant role in controlling several of these reactions. The main aim is to restore the calcium level rapidly to be ready for photon arrival. Lastly, we saw how the retinal molecule was recycled. It started in the 11-cis form, changing to the all-trans form when it absorbed a photon of light. The all-trans form had to go to the RPE cell to be changed back to 11-cis form, then back to the photoreceptor, where it could rejoin the opsin molecule ready for the next photon of light. Phototransduction is an amazing process, both for the function it performs, sensing light, but also for its level of complexity. Here is a set of selected references in case you want to read further.